Welcome back, everyone. We just had an interesting discussion about the role of cyclin-dependent kinases, how they activate and deactivate target proteins in a cell, and how they are activated and deactivated so that the cell is able to carefully go through the cell cycle. It's very important that the cell is able to regulate how it goes through the cell cycle, that it makes sure that it finishes one step before it moves to the next, and that it's going in the correct order so that you end up with normal DNA in those daughter cells. One example of a protein that could result if there was a problem is aneuploidy. Aneuploidy means you have the wrong number of chromosomes in that cell, perhaps too many, perhaps too few. Both types of aneuploidy, too many or too few chromosomes, can cause problems in that developing cell or in a developing fetus if it happened in a germ cell. So cells use a series of checkpoint mechanisms, things that we'll see some regulation by Cdk-cyclinases, but we'll also see some more molecules being involved than just our Cdk-cyclin complexes. So this is so that your cell makes sure it has completed one phase and it's been completed properly before it starts the next phase. One thing that Cdk-cyclin can activate is the RB protein. So the G1 Cdk-cyclin is able to phosphorylate its target RB. When RB is not phosphorylated, it is bound to this transcription factor E2F. But when this G1 Cdk-cyclin phosphorylates RB, it releases the RB. That means that E2F is left by itself to do what it really wants to do. And what it really wants to do is transcribe genes. So E2F will transcribe genes that create enzymes needed to go through DNA replication. So here the E2F bound to RB, no synthesis of proteins for DNA replication. RB phosphorylated, E2F will synthesize those or transcribe the genes involved in DNA replication. Okay, so again, when RB is bound to E2F, the E2F molecule is inactive, the cell cannot enter S phase. When cells are stimulated to divide by growth factors, the G1 Cdk-cyclin is activated and phosphorylates RB. Phosphorylated RB releases E2F, which allows transcription of genes needed for entry into S phase. Another thing that happens is we have to make sure that we replicate DNA. We only want to replicate it one time. You don't want to keep replicating DNA after that. To make sure that you replicate it one time and then stop, we have a process called licensing. MCM proteins are proteins that are going to be involved in binding to the origins of DNA replication. And they're going to be kind of regulated by these two proteins, ORC and by helicase loaders. So here you can see, here's your DNA, starting G1, your double-stranded DNA. And then in G1, you can have ORC, MCM, and helicase loaders bind to it. So this is going to help those MCM proteins bind to their proper place. Then when it's ready for synthesis, we're going to take off ORC and the helicase loaders. That allows those MCM proteins to initiate replication from those origins of replication. Then, to prevent the replication from happening again, MCM gets knocked off. So MCM is stopped from binding to the origins again. This DNA is not going to be able to replicate again. It's been licensed and now that's it. It won't be able to be active again until the licensing system is active for activates in G1 again. Cdk is a player here that will stop relicensing by inhibiting proteins like ORC and helicase loaders. So here you are inhibiting this licensing system because of these Cdk blocking ORC and helicase loaders. Another inhibitor of relicensing is geminin. Geminin is a protein that blocks MCM binding to DNA. So it is going to block this MCM binding here. And geminin is going to be degraded at the end of mitosis so that licensing is able to happen again in the next cell cycle. At the G2 to M transition, we have a DNA replication checkpoint. The DNA replication checkpoint is going to check that DNA has been replicated. It makes sure that DNA synthesis is complete. That DNA synthesis has to be complete before you exit G2 and start mitosis, or you will have incomplete copies of your DNA. So we have cells, and you can prevent them from completing their DNA replication. They will fail to do their final dephosphorylation step to activate mitotic Cdk-cyclin. So that final dephosphorylation we talked about that is required, it won't happen. Cdk-cyclin, the mitotic Cdk-cyclin, won't be activated, and you can't go through mitosis because DNA replication hasn't happened yet. Once DNA replication does occur, then that dephosphorylation step happens, and CDK is active. You go through the steps of mitosis. Mad and Bub proteins are proteins involved in assembling the spindle assembly and checking that the spindle assembly has been correctly assembled. The mitotic spindle checkpoint will stop anaphase from beginning until all the chromosomes are attached to the spindle. Your cell will hang out in metaphase until all of those chromosomes are attached to the spindle. So if there are kinetochores that are unattached, they send a wait signal that inhibits the anaphase promoting complex. That involves these Mad and Bub proteins. They can bind to the chromosomes that are not attached to kinetochores. They will, then when they're released, they bind to CDK or CDC20, which activates the anaphase promoting complex. If they're still attached to the kinetochores, they won't bind to CDC20, and you have an inactive anaphase promoting complex. Okay, so Mad and Bub proteins accumulate at unattached kinetochores. They become part of a multi-protein complex that inhibits the anaphase promoting complex by blocking the action of CDC20. Once all the chromosomes are attached, Mad and Bub no longer persist at kinetochores, and inhibition is lifted. Another checkpoint we have is through DNA damage with our good old P53, my favorite gene we talk about. There are multiple series of DNA damage checkpoints. They're going to look for DNA damage and will halt the cell cycle. They check for DNA damage at late G1, at S, and at G2. And those checkpoints are going to inhibit different Cdk-cyclin complexes. And this involves P53, that good old guardian of the genome. The way that P53 responds to breaks is that when there is a DNA damage, especially if it's a double-stranded break, it activates the ATM, or ataxid telangiectasia mutated protein kinase. The ATM will phosphorylate checkpoint kinases, and checkpoint kinases phosphorylate P53. Phosphorylated P53 can't bind to MDM, so MDM marks P53 for destruction. When phosphorylated P53 is protected from degradation, it can activate two events. One is arresting the cell cycle, and one is cell death. So P53 is going to activate the gene for P21 that inhibits the cell cycle progression. It will inhibit different CDK-cyclins. And then ATR, which means ATM related, will react in response to single-stranded DNA breaks. Okay, so P53 is activated. It will halt the cell cycle. When P53 is deactivated, the cell cycle is able to progress. So P53 is going to stimulate production of enzymes involved in DNA repair. So we are going to activate these things, but if the damage cannot be repaired, if we can't go through these, P53 will trigger those genes for the cell death, for apoptosis to occur. This involves a protein called PUMA, or P53 upregulated modulator of apoptosis, which will inactivate BCL2, which inhibits apoptosis. So we have DNA damage that is going to activate ATM or ATR, depending on if it is single or double-stranded damage. That activates checkpoint kinases. Checkpoint kinases will separate MDM and P53. If MDM and P53 are together, they're degraded and the cell cycle continues. But if there is this DNA damage, if this pathway has been activated, the MGM lets go and P53 can start doing its work. It can either go through P21, inhibiting Cdk-cyclin, so you arrest your cell cycle, or it can go through PUMA, which inhibits BCL2 and leads to cell death. Okay, so that is our pathway, our two separate pathways that P53 influences, either allowing cell cycle arrest so the DNA can be fixed, or if the DNA can't be fixed, going through apoptosis. All right, we're almost done with this chapter. We have talked about a lot of things that slow down and regulate our cell cycle. We still need to talk about how different growth factors will trigger a cell to proliferate. And then we're going to talk about what causes a cell to go through apoptosis. If the cell cycle isn't going to happen and the cell dies, how does it go through those stages of apoptosis? Okay, I'll see you later. Let me know if you have questions and we can talk about it. Bye.